Okay, so while we get settled here, so my name is Chris Hackley. I'm the co-founder and CEO of CRISPR Therapeutics. I'm going to do my best to keep us on time as well. And like Ranjan mentioned, this is going to be a pretty broad overview, right? So we're going for more breadth rather than depth, but I will tell you about some of the exciting stuff that we've been working on and kind of my views of the CRISPR system. Obviously, we're in the Bay Area, right? There are a ton of CRISPR luminaries here. So consider this, a mix of, the hard published science and then what I think is the future, right? So to start, where do we start from? So I think a lot of you are familiar with the fact that around 2000, we were able to sequence the human genome for the first time. Why does this matter? CRISPR is fundamentally a tool used to edit the genome. If you don't know what you're editing, you can't do anything, right? And so this was a big breakthrough. And so one of the most exciting things about this, and as the years have gone on, right, is that we know right now over the exact genetic causes of over 600 or 6,000 genetic diseases, right? We know exactly what the changes in the nucleotides are that are causing these diseases. The issue is that the tools to actually fix them have lagged behind. Okay, whoops, this way. Okay. And so what was so exciting about CRISPR is that when it came out, is it wasn't the first tool that you could use to go and cut DNA, right? So all I'm showing you here, these are basically different enzymes that you can use to cut DNA. The big difference here is that all of them required recoding the protein. And one thing I want you to take away, and you probably already know, based on if you've done any investment in the space, proteins are hard and they're expensive because of that, and they're also valuable because of that, right? Yeah. What makes CRISPR so exciting is that you can actually use it just like Control F in a Word doc, right, to go zoom around the genome. But instead of having to recode the protein each time, like these other tools had to do, which is hard, right? You just replace the RNA, and nucleic acids are easy. I mean, easy, right? So that's why everybody's so excited about CRISPR. If you ever wondered why, right? There are other tools. That's the key, is that you can use nucleic acid instead of having to recode protein. So if you don't know what CRISPR is, just a little bit of review here. Essentially, it is the bacterial immune system. And so what bacteria do, they get attacked by phages all the time, right? What CRISPR actually does is it's just a sequence pattern. So it's literally just looking for a certain sequence of nucleotides that it recognizes. It uses basically some pretty cool ways to record that information in the genome so the E. coli actually can learn. So it's called adaptive over time, right? And adapt to new phages coming in and trying to kill them. And so this is what Doudna and Feng Zhang are famous for, basically, is taking this system and putting it actually in human cells, right? So one of the biggest things that happened for us, and something I noticed talking to pharma partners, is last year there was this first clinical trial that came out, and I know there's a ton of data on here, right? I really want you to look at the bottom here. So this is a sickle cell clinical trial, and what you're seeing here is basically the patients had to undergo infusions or they had nasty side effects, right? Right, where they actually edited the patient's cells to restore hemoglobin functionality, right? Look, they didn't have to do that. So in one treatment, it's cured. And the follow-up data has been equally impressive. And I think this fundamentally changed, at least in my opinion, how large pharma looked at it. And because large pharma has the cash, right, that has fundamentally changed, I think, the landscape of CRISPR. And then there's this other clinical trial that's gone on as well. It showed similar results. Now, the catch is that these are really cool, right? But when you think about CRISPR editing, there's not just, it's really the Word doc, and we'll get to it in a second, is a great analogy for it. You can delete, you can insert, or you can rewrite, right? I mean, just the exact same thing. And basically part of what has held CRISPR back so far, because it's been 10 years, right, since it really became discovered. In theory, there's been who knows how many billions of dollars poured into how many companies, right? We have a couple really exciting clinical trials, but the vast majority of rare diseases. And the reason is that right now we're more or less restricted to using CRISPR in an informationally destructive manner. So you go in the genome, and again, like your Word doc, you've got your cursor, right? You found where you want to edit, you know where you want to edit. The problem is you can't change it to what you want. You can basically just make typos. And that's really useful for some things, like these clinical trials that I just showed you in particular. They can do these workarounds that essentially allow making typos. As you might expect, most diseases, you can't fix them by breaking something else. It really is. It's that simple at the fundamental thing. So there are some new companies that I'll talk about, including, of course, us, that are trying to go at it and get away from the destruction, and instead, let's get to the ball, right? So that's shifting forward. What I think you're going to see in the future of CRISPR is moving towards this ability to really intelligently rewrite. And one day, ideally, it would be as easy as word processor, and put that out, and boom, you can do the editing. So, all right, a lot going on this graph, right? What I want you to take away is that over 10 years, no pop quiz, I'm not going to ask you what each one does. What I want you to take away, right, is we started with one enzyme, right? And in 10 years, there is all this crap, and this is not all of it by any means. So there's just been an explosion in what you can do with it, right? So we started with one thing that I told you could cut and make typos. And now we have all these different things where you can rewrite certain bases. You can use them to turn genes on, turn genes off. The most, I think, prime, for those of you who are following the news, they just IPO'd in this really god-awful market, right? And I'll tell you a lot about their technology because we think we complement them in a very but yes, this is the exciting part, is that there's just been an explosion of what you can do with genome engineering. And so this is actually taken directly from Prime's S1 because I think it is a really great way to understand what you can do at a functional level, right? So here we have a section of the United States Constitution. You see there are some typos in there, right? And so again, it really is like this. So with using the standard informationally destructive way, right? You make typos. So if there are typos in there and you need to fix them, can't do it. You can break stuff. There's this technology called base editing where you can take certain DNA letters and change them into others. It's only a single letter though at a time, right? And then finally you have Prime editing, which is the company that I just mentioned that went public. This is the one that's probably going to draw the most attention and you're going to see mentioned over and over the next couple of years. And this one, what it can do, what their big innovation is that essentially take the equivalent of about half a sentence, and they're able to do not only just substitutions, but they can insert, right? So it's true rewriting. The problem is that half a sentence in terms of the human genome is not that big. The human genome is huge, right? Genes are huge. And so this editing window, while useful, does limit you. So the fundamental idea here is that different patients, that basically a single disease can be caused by a bunch of different mutations, right? So it's not, so sickle cell is actually the outlier where you have one mutation where a ton of patients have it. And this is by and large what people have gone after, these diseases with hotspots. In reality, most diseases have, I heard somebody else say it and I'll totally steal it, these long tails where there are just tons of different mutations that all lead to the same disease. So the idea is that could you make one editor, right, that will treat a whole bunch of different people, and that reduces the regulatory burden, it increases the chances of it actually working, and you actually benefit more people, right? You're not just limited to the only hotspots. So one of the key things that they claim in this, and you're probably getting where I'm going to go here as I'm talking about edit size, right, is that their editor can do edits about up to 100 base pairs. And so that this is their rewrite window, right? And so the literature so far has said if you use this other technique that our company is named after called homologous recombination, or in this case, it's homology-directed repair, to do rewriting, you can only do it supposedly for a really short window, like 10, 12 bases. That's not true. And I'll show you that. But functionally, right? So again, I told you you could do about half a sentence of the Constitution using prime. What if you could do pages, right? Fundamentally changes what you can do in terms of making a therapeutic. And so the other thing that's really important with CRISPR editing is that there are these toxicity events that happen, right? So it's not just that you can make the rewrites, but can you do it safely? And that is a big challenge. Um, and so one of the ways we really wanted to look at is, can you do it? And it's again using this thing called homologous recombination. And essentially, it's just you're providing essentially a Word doc, another Word doc open with a paragraph, sentence, whatever you want it to be, right? You have this information, and you're literally just using Control F. You have your editing window exactly the same. You've selected with a cursor. You replace it, right? So you can rewrite a letter, rewrite a sentence, you can rewrite. And the issue that it's limited. Why, this has been known about for a while. Why hasn't it been used, right? Why have I been telling you this destructive way is being used? Is because the cells dramatically prefer to use this way. So what we wanted to do as a company is figure out, all right, because we know HDR in theory, this editing style that we want to use could do these long range edits, right? We believe that's possible. But we believed we also needed to improve Cas9. And so essentially what we did, don't need to dive in too much on this, but the key I want you to take away is that we provided extra information to the cells using a fusion protein strategy to essentially tell them, so I'll show you a bit of data here. Essentially, the first thing we did is we tried to insert DNA. It's a really simple readout. In this case, we use what's called a transgene. Basically, what we're doing is we're giving the cells instructions to make a certain protein that they don't normally make. And it's super easy for us to tell, did you get the DNA inserted? Yes or no, right? And so what we've seen is that this strategy works. Okay, it increases insertion rates up to 4 times the baseline. We think this really matters, again, because it's an efficiency problem as well as an editing window problem, right? Number two, what we've done is that we've seen that this is our own safety data, but one cool thing about changing the way the cells handle these breaks is that you can actually increase the safetiness, decrease the side effects, essentially, of this editing by sometimes it goes down to 0 or baseline, basically, just can't tell. And that's a pretty fundamental change in thinking about it. It's been thought before that if you create a double-strand break, you're stuck with the side effects, essentially, right? And what we've shown here and with other data that we published is that that's not the case. And we think that's really exciting going forward, and that it changes how you think about right? So part of my rebuttal to Prime's S1, right, is that no, we can get rid of a lot of these. You are not stuck with these effects that they claim you're stuck with, right? If you choose to not use it. And then finally, so this is the newest data that we have. We got this when we were in the Illumina Accelerator. And so remember when you're going back, remember how people told you there was that 10 base pair rewrite window? So here I'm showing you, this is the base, this is the location, just arbitrary, right? And again, this is our doc number 2, the information we're taking and pasting into the genome, right? And then this is the nucleotide that we're changing it to, and I'm showing you the basically number of edits roughly that we're making out of the total percentage of reads that we get. And essentially, we can do over 3,000 base pairs that we're showing you here. And in another experiment, we've gone up to 4,000, right? And so this is 400x what theoretically was possible, right? In terms of prime, we're 40 or 50 times higher. And the key here, one of the thoughts was that you could never get this editing to be consistent, right, all the way through, because it's a huge range. And we've showed, I was actually really surprised when I saw this data, because I was expecting a big U, essentially, where the middle might drop out. And it looks like it's not the case. So we are super excited that we think this technique is truly viable. 5 minutes. So I think for us, the next steps obviously is if you see these numbers here, just totally honestly, the efficiency is low, right? It's our proof of principle experiments. We're working on optimizing it right now. We already have some early returns that it's possible. So we're already 4xing these results just by doing some really basic stuff, and we think we have some really smart things. So essentially, what you see here is that we're using our special engineered platform versus. Interestingly, they both work, but what you can see is that one of the advantages of our platform, again, is pretty, a lot of information. What I want you to see is that there's more orange over here, indels. By and large, for this type of editing, we don't want indels. So essentially, we can do the editing while reducing indel. And what are indels? Indels are these typos, right? So essentially, you don't always get 100% going to the copy and paste or the typo way. So essentially we can reduce the typos while still keeping the levels of the long range gene writing. And so again, like, why does this actually matter? Why am I harping so much about these large editing windows, right? And it's because human genes are huge. I don't know how many of you have looked at gene models, but essentially just to go over it here, these little vertical bars are exons. These are the things that actually code for the proteins. What you can look for here, these are mutations that are reported to this service called Funbar. And these are mutations that basically people are diagnosed with, in this case, hemophilia. And they've actually been sequenced and logged by clinicians saying, all right, this patient has hemophilia, this mutation has been seen. We submit it, right? So it tells you roughly where the mutations are and roughly the patient, the amount of patients that have it. And what I want you to see here, right, is this is an example of a gene. It doesn't have a hotspot. As an example, cystic fibrosis is a gene with a hotspot. 85% of patients allele, right? This one, there's no allele that's above 1%, right? So fundamentally, what this allows us to do, so this region that I showed you we can rewrite is actually this exact same region blown up here. There are over 65 individual mutations that have just been recorded here. This makes up roughly 20% of the patient population. And I mean, you can probably see that I know these numbers are huge, right? But the distances here are big. This is 3,000 base pairs plus, right, just here. And so essentially what I think this does, it not only will it allow you to develop best-in-class therapeutics, it makes these diseases with long tails much more viable. And I have to be totally honest, I'm giving Prime much more benefit of the doubt. If you read their S1, they're still going after hotspot stuff. But here I'm just showing you basically the amount of patients you could theoretically treat with their editing windows, and then some very back of the envelope math. But what you can see, right, is that it really does take some of these ones that are kind of make them theoretically worthwhile. So that's where we think our leverage is these ability to do these long rewrites. We're super excited about it. We're talking to a bunch of pharma players, about how we can collaborate, because we just want to go, right? I've wanted to know the answer to this question for a really long time. And I'm excited to say we can do it. So I'm really excited to see what happens.
And talk about rise minutes. Yeah, so by the way,